Natalia Z, and today I will present my article, which is the protective role of ferulic acid against cisplatin induced ototoxicity. First of all, I'm going to start talking about the main compounds used in the research. So we have the cisplatin, which is a chemotherapeutic agent used to treat head and neck tumors, testicular and ovarian cancers, osteosarcoma, medulloblastoma, neuroblastoma, Hodgkin and non-Hodgkin lymphomas. It works by halting DNA division by making cross-links between DNA chains and thus inhibiting the polymerases during the S phase and thus preventing the cell to replicate damaged DNA. Also, its toxicity is shown as leukopenia, nephrotoxicity, neuropathy, and as we will see, ototoxicity. And we have the ferulic acid, which is a phenolic compound that can be found in wheat, rice, and peanuts and has antioxidative properties as a preventive of oxidative stress through free radical scavenging and lipid peroxidation. It also has properties proved as anti-cancerogen, cardioprotective, hepatoprotective, and neuroprotective. So, the main mechanism of action of the cisplatin is by increasing ROS, not just in tumoral cells but in normal cells, including hearing cells. But the problem is that the increase of lipid peroxidation in the cell also has like a decrease of antioxidant levels and an increase of calcium inflow into the cells, that all that leads the cell to apoptosis, and that can be shown as hearing loss and tinnitus that can or not improve after the treatment. The objective of the article is to investigate the protective effects of ferulic acid against cisplatin-induced ototoxicity. Now I'm going to start describing the methods used in the research. So first we have the tunnel assay that is based on the apoptosis, where the cell goes through DNA fragmentation, and each of those DNA species has a 3-hydroxyl site that can be tagged by an enzyme called TDT, terminal deoxynucleotide transferase, that also can be fluorescent labeled and then detected by fluorescent microscopy, and that helps us to determine the cells that are going through apoptosis path. Also, we have the real-time PCR, which is a technique that allows us to create like DNA replications of a gene fragment. In the research, they were made from 4 antioxidant enzymes: the glutathione peroxidase, glutamate cysteine ligase, superoxide bismutase, and the catalase. So, the antioxidant enzymes are the main line of defense against ROS. In the research, cells were first harvested, then their RNA collected and reverse transcribed into cDNA, complementary DNA chains that were used to make the replications of the genes. So this technique was applied in the research to determine the mRNA expression, because the cisplatin will reduce them because it reduces the synthesis of proteins. Another assay performed in the research is the Western blot, which is an assay that is used to separate a specific protein from a mixture of them extracted from the cell by gel electrophoresis. Gel electrophoresis separates proteins by weight and charge. So in the gel electrophoresis we have like a blood tank, and we have a gel and a membrane, and we put the proteins on the negative side because we have like an electrode, because we are going to induce electricity through the sample. So the proteins will travel a certain distance from the negative side through the positive side, and then we will target them with specific antibodies that then are going to be detected by a Western blot detection system. And this assay was made for check the levels of cleaved caspases, cleaved PARPs, NRF2, and catalases, because cleaved caspases are active caspases that show that the cell is going through apoptosis. Cleaved PARPs are proteins that normally repair the DNA from the cell, but when they are cleaved, they are like corpses, so the cell is going through apoptosis. And the catalase is an antioxidant enzyme by itself, but the NRF2 factor is a transcriptional factor from other antioxidant enzymes. And the last assay is the measurement of ROS by flow cytometry. So for that, we use cells that were harvested with mitosocs, which is a dye that selectively targets the mitochondria. And then when the cell has like oxidative stress, in this case superoxide, which is the main ROS that affects the mitochondria, it will turn fluorescent and can be detected by flow cytometry in this case. For the flow cytometry, we put the sample in a transparent tube and then a laser that goes through them, and shows like by detection system the population of the cells in the sample. Now I'm going to present the results of the essays. Here we have the Western blot where we are analyzing the levels of cleaved caspase and cleaved PARPs. Here we have 4 groups. On treatment group, cisplatin treatment group, ferulic acid, and cisplatin that was pre-treated with ferulic acid. Here we can see that the untreatment group and the ferulic acid don't have, neither of them, the expression of cleaved caspases or cleaved PARPs because they are like not going through apoptosis. But the cisplatin group has like the higher expression of those molecules, and that expression was significantly reduced by the ferulic acid pre-treatment. Here we can see. And these graphic bars shows like the same thing, that the cleaved caspases and the cleaved PARPs has like the higher expression when they are just with cisplatin, but that expression reduced when they are like pre-treated with ferulic acid. And here we are, is another Western blot for the NRF2 and the cleaved caspases. Here we can see that without ferulic acid but with cisplatin at 8 and 16 hours, the expression of those molecules is like meh, because these are like having a stimuli of the oxidative stress, but those expression is significantly increased when they are treated with ferulic acid, here at 8 and 16 hours. This graphic shows the results of the tunnel assay, where we are searching for apoptotic bodies. So here we can see that positive tunnel cells in the cisplatin group and in the ferulic acid pretreatment, but those expression of apoptotic cells is reduced with the ferulic acid pretreatment. The ferulic acid and the untreatment don't have any of apoptotic bodies. And here we are comparing with another technique, which is the DAPI, that is not specific for apoptosis because it just binds to the nuclei when it is complete. But for example, with the cisplatin, we can see that the nuclei is like fragmented, and that fragmentation is reduced, so the nuclei is more complete with the ferulic acid pretreatment. These are the results for the flow cytometry of the rows of the mitochondria. So here we can see in this axis like a more concentration of those rows, and here like the population of the cells. So here we can see in black the amount of unknown treatment group that has like the highest population, as well as the ferulic acid group. The lowest population is just with the cisplatin, and this population is increased when we have like the ferulic acid pretreatment here in green. And this graphic shows as well the ROS production that is higher with the cisplatin and is reduced with the ferulic acid pretreatment. I have 2 personal conclusions of the research. Further, the ferulic acid has shown properties against cisplatin ototoxicity, but it should be more used and studied in treatments and prevention from other diseases to protect patients against ROS. And also here it's really important the molecular biology that allows us to understand, for example, that the NRF2 is a transcriptional factor which upregulates the expression of another antioxidant enzymes to like counteract those ROS, and that give us the opportunity to design new therapeutic methods to fight against ROS. This is a discussion where we are comparing like our results with other author results. A first author said that the currently accepted mechanism of cisplatin-induced ototoxicity is by generating oxidative stress by ROS, and that also agree with our results. Another author said that cisplatin cytotoxicity can be counteracted by endogenous defense system as antioxidant enzymes, and that also is current with our results. And a final author said that ferulic acid show protective effects against cisplatin cytotoxicity in auditory cells, which is like the same objective of our result. Finally, this chart is like the summary of the main points of the research. So it starts with the cisplatin that generates ototoxicity that can be shown as tinnitus and hearing loss, and is caused by cisplatin mechanism of ROS production. The cisplatin is a chemotherapeutic agent that halts cell division during the S phase by generating DNA damages. That's why it can lead to a secondary cancer and also to apoptosis into normal and tumoral cells, for example, hearing cells, and causing ototoxicity. The DNA damage is caused by generating ROS, and ROS are molecules that overload cell antioxidative systems by leading to a biological damage, for example, lipid peroxidation, protein and DNA damage, abnormal behavior, for example, accelerated senescence, abnormal proliferation, or dysregulated inflammatory response. Those damages can be counteracted with antioxidants, for example, the ferulic acid, which is an antioxidant compound that also has antioxidative properties like anti-carcinogenic, cardioprotective, hepatoprotective, and neuroprotective. It works by increasing antioxidative cell elements and reducing growth, by antioxidative enzymes, and also by the NRF2, which is a transcriptional factor that increases those enzymes by itself. So that's why the ferulic acid can be used in cisplatin therapy, for example, because it reduces the effects in normal cells. And that's why ferulic acid should be more used in normal life, and also to protect like other patients against some oxidative medical treatments.